Great. Well, thank you for that introduction. I benefit today from a great series on the microbiome yesterday and, of course, the preceding talk. It's great to see a lot of interest in metagenomics and microbiome work at ISMB this year. So I'd like to continue the discussion about our growing understanding of the human gut and the methods we're using to do that. So I'll start a little bit with an introduction on the human gut. This is a quote that I found on Reddit that I really like, that our gut is a microbial mothership, which the reason that that's the case is because it's home to trillions of microbial cells from hundreds of species. And if you go across all of those species, you find about 100 times the gene content of our own genome. And the reason why we know all of this is due in large part to several recent large-scale efforts to map the human microbiome, including the gut microbiome by MetaHit, which we heard about yesterday and today, and also the Human Microbiome Project. And efforts like that have revealed some interesting findings. For example, we know if you look across individuals in the gut, we see a lot of variation in the membership of that community. In this case, a big interchange between two different major phyla there. However, if we look at microbial function in the gut, we see it's much more conserved across individuals, in this case in the sense of KEGG pathways. But I want to emphasize that when we're doing this, when we're doing metagenomics to understand community membership or function, what we're really talking about is functional potential, what the community is capable of doing. And I'd like to contrast that with functional activity, what the community is doing at any particular time or under a particular set of conditions. And that's the sort of thing that we can survey with other technologies, in our case, RNA sequencing, but also proteomics and metabolomics. And so we'll talk today about some of the similarities and differences between the functional potential of the human gut microbiome and its functional activity. And to set that up further, this is an idea, this contrast between potential and activity, that we're all very familiar with in the context of multicellular organisms, where, for example, all of these different tissue types or cell types in the human body have identical functional potential. They all have the same genome, barring some sort of unusual event, but we know that they have very different functional activity. What genes are on and off at a particular time are very different. And so the question is, to what extent are these properties, activity and potential, different or the same in the microbiome? So to do that, we use a combined metatranscriptomic RNA sequencing and DNA sequencing approach. We had 8 adult male volunteers from the local health professionals follow-up study cohort, and they provided samples that we actually processed by a number of methods, which I'll talk about at the end if there's time. And from these, we did a shotgun sequencing approach to get this DNA confetti. I liked that term from the previous talk, and then sequenced it by Illumina HiSeq. And then those reads are quality trimmed and the host reads are removed. So we have to remove the human DNA that could be in there. For the exact same samples, we do RNA sequencing, where we take out the RNA confetti. And then there's one special step that we have to do when we're doing RNA-seq on microbial communities, and that's to experimentally deplete the 16S ribosomal RNA. If you don't do this, then about 90% of the reads that you get are from this one gene, so just an interesting fact there. But then otherwise, the sequencing procedures proceed much as they do for DNA. We do a conversion process to convert these RNA reads into cDNAs, and then they proceed along the same pipeline that we saw above. And we refer to this top part here when we're focusing on DNA as metagenomics, the right-hand part as metatranscriptomics, and collectively we collapse these together into something called metaomics. And so you'll see the word metaomic in the title of this slide, for example. That's sort of some of the experimental and sequencing aspects of the study. In terms of the bioinformatics, since we have these reads, what do we do with them? Well, we're interested in function, for one. And so the way that we do our functional profiling is by doing translated search of these reads against protein families, in this case against KEGG ortho groups. And so this gives us a sense of sort of the discrete units of function that we find in the community. And this works either with the DNA or the RNA. And then these can also be further hierarchically summed into pathways and larger functional units. So that tells us what the bugs are doing, and that's all done with a tool called Human that's available through the lab's website. To find out who's there, what the actual bugs are, we use another pipeline. It's again a mapping step, but now a mapping against specific marker genes for species in the community. So for example, if we have isolate genomes from this purple bug, we specifically map the reads to these marker genes for that species that are always observed there but never observed anywhere else. And we use this to get an accurate profile of the species composition of the same samples. And this is done with a tool called Metaflan that's also available. And both of these tools give us profiles like this, sort of a table of samples by features where the features are either functions or species. So now we have the data, what can we do with it? Well, the question that we really set out to answer first and foremost was whether functional potential and functional activity are the same. So those profiles give us a picture like this when we're looking at DNA information. We have, in this case, 3 functions with increasing potential, 1 copy of gene A, 2 of B, and 3 of C. When we do the transcriptomics, we get a sense of functional activity, what the cells are actually doing at a particular time. If potential and activity are the same, then we'll end up seeing a picture like this, where the relative abundance of each of these functions will be the same at the RNA and the DNA levels. If we were to plot them against each other, we would see a nice line, barring for noise and so forth. And so that's our question. Does potential equal activity in the human gut? So what do we actually see? Well, if we do that plot that I just talked about, we see a surprisingly strong correlation. This is not what you would see if you were looking at a human cell type, where many of these genes would be off at any given time. And so we actually see here that as the number of gene copies of a particular function increases, we tend to see a correlated increase in the number of transcript copies for that gene. And so this is telling us that, unlike the human genome, these microbial communities are actually exercising a lot of their potential, at least in this healthy gut situation. However, you can see that there's a lot of variation around this trend. So what we're also interested to know is, are there functions that disobey this rule? And so what we can do is use a gene set enrichment-like approach to try to find functional categories that are either enriched below this line, meaning that there's more DNA than RNA, they seem to be underexpressed, or functions that are enriched above the line. We're seeing more RNA than DNA, and this suggests that they seem to be overexpressed. And so, for example, here is a first example of an underexpressed function, the biosynthesis of these 3 amino acids. And this was actually a common trend in the data set, that biosynthesis of small molecules tended to be underexpressed relative to their genomic potential. And this seems to make a lot of sense when you think about the healthy human gut, which has the remnants of our diet in it, a lot of food particles left over. All of these compounds, these small compounds, will have high bioavailability. And so it makes a lot of sense that the microbes, if they can just import those compounds, would choose to do so rather than synthesizing them de novo. Another function that we saw is really strongly underexpressed was sporulation. So sporulation is a stress response that's used by certain gram-positive bacteria, and so we would not expect to see this as an active process in healthy individuals, as these are. But yet it remains in their reservoir, so it's actually there kind of sinister, sitting around waiting potentially to cause a problem under stress, as in the case of C. diff infection. But we see in these healthy individuals that it's very strongly underexpressed. Most of the genes are not really being utilized. So that's underexpression. On the other side, we see functions that are much more highly expressed than we would have expected from their genomic potential, metagenomic potential. This is very much a positive control here. We sort of know from the biology that ribosomes are synthesized at a very high rate, and so we would expect to see the ribosomal protein coding genes to be highly transcribed, and indeed we do. You can see, in fact, for 3 different domains of life, bacteria, archaea, and eukaryotes, we can consistently see that their ribosomal protein coding genes are being more highly transcribed than we would have expected from their gene copy number. What's especially exciting here is that eukaryotes are frequently undersampled in metagenome just because they're small in abundance relative to the bacteria, but they're making enough of their ribosomes that we can actually start to see them peeking out here in the metatranscriptomic data, which is very cool. Another function that we saw is very highly expressed was methanogenesis, and this was more of a surprise. If you actually flip back and forth between these 2 slides, you can see that methanogenesis is sort of spiking up in the same cluster as the archaeal ribosome, and that actually makes a lot of sense because methanogenesis is a process that's carried out by the archaea that live in our gut. We have a couple of archaeal species that are there along with bacteria, the biggest of which is Methanobrevibacter smithii. And what those species do is they actually take the waste products of bacterial energy production, such as formate, and further reduce them to make methane. And in so doing, they actually drive the energy balance of the gut in a lot of ways. And so what you can actually see happening here is that this is a very active process in this species, which to me makes a lot of sense. If there's just one species here that's doing this function that has an important role in the gut, its metagenomic potential can be relatively low. It's not a very common function across bacteria. In fact, it's in the archaea, not the bacteria, but it's being really highly expressed in the organisms that have it. And lastly, and quite apropos relative to the previous talk, we actually find that the gene for tetracycline resistance, TET-A, is extremely highly expressed across all of these individuals. So this is a case where if you're doing antibiotic profiling or antibiotic resistance profiling using metagenomic data, you're going to tend to underestimate the impact of this gene, because we can actually see here that it's quite highly expressed relative to its metagenomic potential. So in all of those examples, what we were looking at was a community-wide function. We're actually looking at these functions as they are across all species, looking at all gene copies across species and sort of adding them together. There's additional information we can learn by stratifying those functions across the individual species that are doing them. And the example of methanogenesis was a good one, where we can see that in that case it's very obvious that that's a function that's being done by just a single species, because there's actually only one species that's commonly in the gut that can do it. But for some of these functions, they're broadly distributed across species. And so when we do a community-wide profile, we're actually seeing the sum across all of those different signals. And so one example of that, which was sort of interesting to us, is, as I mentioned before, we saw that ribosomal proteins across the 3 different domains are being highly transcribed. But peptidoglycan biosynthesis was one of the underexpressed pathways. We didn't look at this one yet, but it was one of the more significant ones from the list. And this is puzzling because both of these processes are associated with bacterial cell growth. So you expect that cells that are more actively growing are making more ribosomes, and they're also synthesizing more cell wall, more peptidoglycans. So why are these trends going in opposite directions? And so this was something we were able to answer by stratifying these enrichments by species. And what we saw was a picture like this. So in this case, what we have are the bacterial ribosomal protein biosynthesis on the x-axis and the peptidoglycan biosynthesis on the y-axis. And what we're looking here is the fold transcription over the fold metatranscriptomic abundance, RNA, over DNA abundance. So for example, if you're a 2 here, it means you were 100 times more abundant at the RNA level than you were at the DNA level. And that's for this whole pathway. And then each of these dots here represents an individual species contribution to these pathways. And so what do we see? Well, here it's very clear that the 2 functions are correlated, as we expected. As you're doing more ribosome biogenesis, you're also doing more peptidoglycan biosynthesis, which is what we expected from the biology. However, it's not a paradox, because what we actually end up finding is that for the majority of species, they're enriched for ribosomal protein transcription. You can see that most of the species here are in the positive for ribosomal protein transcription. In fact, it's about a 4-to-1 ratio of species that are in the positive here to the negative. So it looks like, consistent with what we're seeing here, most species are over-transcribing that function. Whereas for peptidoglycan biosynthesis, we see a 3-to-1 trend in the other direction, that most of the species here are actually not synthesizing as much peptidoglycan generating enzyme, or transcribing as much enzyme, as we would have expected from its metagenomic abundance. So everything here checks out, but it requires taking that community level function and stratifying it by species to understand what's going on. So not a paradox that's consistent with the biology. And in this case, what's probably going on, this is our hypothesis anyway, is that ribosomal proteins have to be continually synthesized. The byproducts of this transcription are actually physical building blocks of the ribosome. Whereas in the case of peptidoglycan biosynthesis, what that pathway is actually making are the enzymes that go and build and refine the cell wall. So it makes some sense that perhaps you don't need quite as many of those as you would the actual ribosomal components. And so it all seems to check out. Okay. But in everything we've looked at up to this point, we've been averaging over those 8 individuals. Another big question that we're interested in the context of functional potential versus activity is whether you can have different functional activity in the context of a conserved functional potential. That's exactly the case across human cell types, for example, where they all have the same functional potential but very different functional activity. So what we found is that our data actually recapitulate one of the main figures from the Human Microbiome Project paper, which is that if you look at species or genus level abundance across the 8 different individuals, you see quite a bit of variation. People have very different compositions with respect to these 10 major genera. However, if you look at function, functional potential, so looking at functions at the DNA level, it's much more conserved. And this is now a result that seems to show up over and over again when we look at microbial communities, that there's something about the gut environment that imposes a certain set of selective pressures on the community, and so that species that can actually do this set of functions do well in the gut, but that there are multiple sets of species that can sort of solve that problem of how do you get the sets of functions. So what does this look like at the RNA level? Well, it's somewhere in the middle. We see that it's not nearly as different between individuals as, say, taxonomic composition, but it's more, the RNA expression is more variable across people than the functional potential measured with DNA. And if we do a measure of ecological similarity, like Bray-Curtis score, you can see that same trend here with more statistical oomph behind it, that the genera are very different between people. Functional potential is very well conserved, but functional activity is very unique from one person to another. And so we can actually dig into this more and see what's going on, which functions are being tuned in this manner. And so here's one example where these bacterial ribosomal genes are extraordinarily well conserved across people. And this is what we expect for genes that are really well conserved across bacteria. If every bacteria brings a copy of this gene to the community, then we expect that everyone will have a fairly even amount of it as we go across communities and bugs and people. However, you can see very strongly that there's a difference in transcription across these individuals, which might correlate with something like growth, that the community in this individual here might be growing less than the community up there. And this is one of the few slides in this talk that's not on a log scale. So you can see these are actually very, very similar, and these are quite spread out relative to them. And if you actually dig into this further, what you tend to see is that these are all from the same individual, so that this cassette of genes is being consistently upregulated in this individual relative to this individual down here. And these patterns also tend to be very consistent across the 3 technical replicates that we have, or the 3 different sampling methods, I should say. So it doesn't seem like it's just an issue of, say, RNA being more stochastic than DNA. So conclusions. We saw that functional potential and functional activity are actually quite well correlated in the human gut. And this makes them very different from something like human tissue types. And so this is actually great news for people who are doing functional surveys based on DNA, because it tells you that if you're measuring DNA, you're directly measuring functional potential, but you can make a very good guess about the functional activity in most cases of the community. However, there are these sort of second-order effects of things that are more abundant at the RNA level or less abundant at the RNA level than you would have predicted based on DNA alone. And tetracycline resistance was a great example of that, where here's this function that we can see consistently across individuals at a relatively low metagenomic abundance, but it's being really highly upregulated. And lastly, we saw that functional activity could be fine-tuned in a background of constant functional potential. So we had this situation where, as we go from person to person, we saw very similar composition in terms of what functions they actually had that was well conserved, the potential was well conserved, but we see this fine-tuning, that some people are doing some functions a little more and some a little less. Now, to fully understand what's going on there, you really want to have time series data. So you can ask 2 important questions, one of which is, are those changes stable over time, or are those differences stable over time? Is my microbiome consistently upregulating a function relative to your microbiome? Or alternatively, something that you could see with time series data is whether those changes, the changes in the functional activity, are the result of some stimulus that we can measure. Is my microbiome doing something a little different than yours because I just ate a big sandwich, for example? Something like that. In a way that you're seeing sort of a short-term burst of activity that doesn't end up going on to change the overall composition of the community. And Peter Turnbaugh had a great example of that yesterday with the xenobiotic degradation, where you have a bug that switches from metabolizing, I think it was arginine, to a xenobiotic, and you can see that at the transcriptional level, even though the bug itself doesn't really change in abundance, it's equally happy to eat either of those compounds. And lastly, for time constraints, I wasn't able to get into all the details of this, but as part of this study, we validated a procedure where if you want to do this kind of meta-omic analysis, combined DNA and RNA sequencing, you can reliably have individuals take a sample at home and mail it to you in a fixative over 48 hours, and that's very consistent with a directly frozen sample, like as was used in the HMP protocol. And so that's really great, and that's going to allow, I hope, a lot more metatranscriptomic work to be done in the future, since you can sample a broader collection range, and it's just much more convenient for all the people involved. If you're interested in knowing more about any of this, in particular that protocol, it's all detailed in the paper that was accepted for this talk. Here as a highlight, and it has the same title as the talk, so it should be easy to find. And if you're interested in any of the tools we used, they're linked in the paper, but you can also check out the Huttenhauer Lab website, which will give you a link here to our Bitbucket repository of all the different tools we have for both functional and taxonomic profiling, as well as the underlying statistical analyses. So a big thanks to the Huttenhauer Lab itself and my mentor, Curtis Huttenhauer, for getting me up to speed on all of this and introducing me to the fascinating world of our microbial motherships and so forth. Several alumni from the lab who were around when I first started and really gave me a good heads up on all of this. Of course, all of the co-authors and collaborators from this project who gave us great feedback and helped with sample recruitment. The volunteers from the HPFS cohort themselves who were nice enough to provide these samples. Our funding sources, and the NSF for supporting my visit to ISMB this year. Thank you.
Very nice work. I'm just wondering, sort of on the practical side of things, since the metatranscriptome and metagenome is more expensive data to collect, like how deeply would you need to sample to sort of get the results that that you showed?
Yeah, good question. So all of the samples had about 2 giganucleotides of total sequence, so about 10 million reads per sample in the end.
Okay, so actually not that much.
Yeah,
not
I mean,
yeah.
basically about the same amount of RNA as we typically do with a metagenome sample. Because you're filtering out the ribosomal RNA in an experimental step, you're going to enrich for some of those other functions. If you don't do that and you just directly sequence, you end up mostly getting RNA. So some experimental work upstream can really save you there.
Great. And then I'll make one other comment, which
Yes.
is that the methanogens
Yes.
are pretty well known to very, very highly express that methanogenesis machinery.
Oh,
so
interesting.
those results are pretty consistent to what's known. And
Fantastic.
Good to see.
Thanks.
Hi, great work. I just had questions about metatranscriptomics
Yes.
in terms of kind of reproducibility of the data, and also in the sense that when you were collecting the samples and you did metagenomics and transcriptomics, did you do it, when you take a stool sample, you just section it off at the stool level, or do you kind of homogenize the whole sample and then extract things on?
So I actually have a nice little follow-up slide here to help with this. So we take the sample, and then the sample is originally delivered to us just as raw sample material from the donors. And then for our case, this was just separately aliquoted into a frozen aliquot, and then these other aliquots that were directly deposited into a fixative, and then they went through this mock shipping pipeline. But you can directly aliquot from the raw sample material into any of these. You don't have to homogenize.
The reason I ask is because people have shown that if you take a stool sample and just take various portions of the stool sample, the community compositions are different.
That's totally true. So I mean, if you want to get a more of an average signal over your stool, you could homogenize it, although it's not necessarily something we recommend, or not something we require as part of this protocol. But you are right that you do see variation throughout the stool.
So this is very cool.
Thanks.
I'm curious, do you want to do the other 2 omics as well in your meta-omics, so metabolomics, metaproteomics?
Why not? So one of the nice things about the metatranscriptomics is it follows really nicely from all the pipelines we have in place for genomics. The samples that are compatible with metagenomics are also compatible with metatranscriptomics, whereas in metaproteomics, for example, you need different starting material. But I'm very curious to see how this sort of difference between activity and potential is reflected in those other signals. We know, for example, that in general, if you look at RNA versus protein, those 2 different measures of activity, they correlate, but it's not amazing, even with, say, within yeast, for example. So I'd be curious to see how the protein signal looks here.
Second question, this is pie crust data, right?
No, this is all shotgun sequencing.
Okay,
So we
then
can
that falls, because otherwise you could have had differences resulting from more carriage of things that would explain.
That's right. Yeah, in this case, though, we have actually the shotgun reads that we can map in the same way, whether they're DNA or RNA.
Third and last question, what system of functional representation do you use? Is this KEGG or?
In this case, we were working with KEGG. So KEGG orthogroups is sort of the quanta of the function, and then their pathways, but we're moving toward using UniRef and Metapsych to replace those two since KEGG is no longer freely available.
Awesome, thanks.
You're welcome.